Okay, I restarted my computer and the software, and hopefully the glitching is over. So let's do the activity that I proposed to you last time. Let's take a look at dopamine from the beginning part of its synthesis all the way to its breakdown. Let's follow its activity. So if we have a axon terminal, right, we know that in this axon terminal we've already created the dopa, and this gets converted to dopamine, right, by the enzyme that I mentioned, dopa decarboxylase. Okay, dopamine is then put into vesicles, right? So we'll use the letter D for dopamine. So dopamine is loaded into vesicles by VMAT, the vesicular monoamine transporter. Okay, so the vesicles are now loaded with dopamine. There's a synaptic release event. Dopamine is released into the synapse, right? And postsynaptically, right, it's going to bind to a G protein coupled receptor, right? That's not an ionotropic receptor, so it doesn't directly open an ion channel. Dopamine binding to this G protein coupled receptor, right, will allow the G protein that's associated with it, right? So there's typically a G protein that's nearby the G protein coupled receptor, and when the ligand binds to the G protein coupled receptor, the actual G protein can go off and have a multitude of effects. So, for example, right, it tends to activate the enzyme adenylyl cyclase. Okay, now adenylyl cyclase has a wide variety of activities that it performs in a cell, so we'll leave it here for now, and we'll come back to the actions of adenylyl cyclase in a further chapter. Now, what happens to the dopamine that's in the synapse? Well, there are two things, right? The first thing is that it can enter back into a neuronal glial cell, right? So say we have a glia over here. So this is a glial cell. And so the dopamine is going to get back into either the presynaptic neuron or the glial cell. Oh boy. And it does this, it enters in here by something called a DAT, a sodium-dependent dopamine cotransporter. So there'll be a DAT here, or a DAT here. And this allows dopamine to get back into one of these cells from the synapse where it was cleared. Okay. Once it's in the cell, the glia or the neuron, right, they contain enzymes, MAOs, monoamine oxidases, or COMTs. These are enzymes, and they help degrade dopamine. So we mentioned that one of the drugs of abuse, cocaine, also amphetamine, blocks these DATs. Right, and by blocking DATs, dopamine remains here longer. We mentioned that early antidepressants inhibit MAOs, again, causing different amounts of dopamine to remain in the synapse longer. So when I say, sketch out the process of dopamine from its synthesis to its release to its degradation, this is what I'm referring to. Before we move on to epinephrine and norepinephrine, I just wanted to point out that I mentioned that initiation of movements is a significant function of dopamine, and it does so via this part of the brain here, right? That's what this image is. Oops, I want to go forward. Let's go back. That is the substantia nigra and the VTA, the ventral tegmental area, right, which are involved in initiation of movements, but all of those more complex functions, those are happening at the level of the cerebral cortex. And so these arrows, I think, are just there to show where all of the dopamine effects, the dopaminergic effects are mediated, and you can see it's really mediated throughout the brain. The next thing we're going to talk about is on the right figure, right, norepinephrine and epinephrine. And these two things are similar, and they thankfully are going to use a lot of the same structures, a lot of the same enzymes as dopamine does. So the two other catecholamines are what we're going to talk about next. So we can use norepinephrine for many excitatory functions. For example, let's say functions here. Whoops, I'm already skipping ahead. regulating wakefulness, attention, even feeding. They're also used in the visceral motor neuron system. So norepinephrine is going to be one of those fight or flight chemicals that's released, one of those fight or flight neurotransmitters released to mediate those types of effects. And we'll talk about those effects when we get to the spinal cord. So the visceral motor neurons use this as part of the sympathetic activity. Oops. NE norepinephrine. For sympathetic type of responses, right? Those are the fight or flight types of responses. So if you're typically secreting digestive juices, the visceral motor neurons will release norepinephrine and inhibit the release of those digestive juices, for example. How about synthesis? Well, thankfully, it's just one more enzymatic reaction from dopamine to get to norepinephrine. One step. Okay, and because of that, right, because they look so similar, it's only one simple reaction, it can use many of the same molecular players, I guess, as dopamine does. So for example, when we think about loading in vesicles, how does norepinephrine get loaded? It uses the same vesicular monoamine transporter. Whoa, buddy. The same VMAT, vesicular monoamine transporter, loads the norepinephrine into vesicles. Degradation. Similarly to dopamine, it's also going to use MAOs and COMTs to break it down. There's a slight difference in how it's cleared. It's a sodium-dependent co-transporter that can also clear dopamine, but it's got a greater affinity, or that it's got a greater ability to bind and bring in norepinephrine, and that's called NET, sodium-dependent co-transporter. So we'll talk about clearance from the synapse. So N sodium, NE norepinephrine transporter. And it's a sodium-dependent transporter that will co-transport it up. The receptors are where we see the majority of differences. So we'll talk about the receptors next. So always G-protein coupled receptors, and there are two types, and beyond that there are subtypes. So there are alpha receptors, and there are beta receptors. Of alpha, there are subtypes, alpha 1 and alpha 2. And with respect to beta receptors, there are 3 subtypes, but we'll really only talk about them, so it is 1. So we'll just write 3 subtypes here. We're not going to get into that level of detail. With respect to alpha 1, it works through the inhibition of a particular potassium channel, which slows depolarization. So when alpha 1 or adrenergic receptors are activated, it slows depolarization by inhibiting a certain type of potassium channel. When alpha 2 receptors are activated, it slows down the hyperpolarization. by inhibiting a different type of potassium channels. This is truly annoying. By inhibiting a different type of K channel. Okay, I mentioned a term that I don't think I talked about, right? Another name for norepinephrine is noradrenaline, right? And I think we did that. And so the receptors, these receptors here, are called adrenergic receptors after adrenaline, right? So the receptors are adrenergic. Adrenergic is what that's supposed to say. So there are alpha adrenergic receptors and beta adrenergic receptors. There are alpha 1 adrenergic receptors and alpha 2 adrenergic receptors. These beta receptors have most effect on smooth and cardiac muscles. So you might have heard people who take a beta blocker, right? A beta blocker is blocking the beta adrenergic receptor such that norepinephrine can't have any influence, okay? And it targets the cardiac muscle there. We'll just mention briefly epinephrine because so much of it is the same as norepinephrine. In order to synthesize it, right? So the synthesis, oh, where'd it go? Is from norepinephrine. So again, in one single enzymatic step, norepi is converted to epi. Okay. In the brain, it's found in much lower levels, so in reduced concentration, suggesting that it may not be as important. So we'll say that as reduced concentrations in the nervous system. Oh. Compared to the other catecholamines, to dopamine and norepinephrine. The same VMATs are used. As far as loading into vesicles, nets can transport so that's the same, and the receptors are the same as they are for norepinephrine. So there's both alpha and beta receptors and subtypes of those. So the last thing I mentioned about norepinephrine and epinephrine is what's over here in this image, right? So here's norepinephrine or epinephrine that binds to the G protein, right? And you can see that that changes the conformation. So in an inactive form, this G protein is sort of loosely associated with the receptor, right? But when it's active, everything kind of comes to the receptor, sort of takes its instructions, and is able to go and have many effects, many different effects in the cell. Finally, what's the effect? Well, we know that these neurons that secrete epinephrine project to areas of the hypothalamus. The hypothalamus in the brain is like the master regulator. It's responsible for a lot of homeostatic drive. The medulla. The medulla is responsible for some basic functions. But what these neurons that are secreting epinephrine, what they're doing in these areas, we don't know, right? The function of these epinephrine secreting neurons is not known.